Ultimately for cancer immunotherapy, what we want to be inducing are CD8 T cells. And we know type 1 interferons and especially interferon alpha are important for that. But we sometimes forget that these T cells evolved for the purpose of killing viral and especially retroviral infected cells. And I think I have come to the conclusion recently that TLR7, 8, 9 have a really critical role in detecting that. And that has implications that I want to spend some time today discussing. So just starting with the central dogma, it's obvious the flow of genetic information. And as soon as life evolved, bacteriophage evolved to try to hack into that flow. And so in order for life to succeed, nucleic acid immunity evolved. And these mechanisms, nucleases, RNA interference, CRISPR-Cas, incredibly powerful mechanisms, these all work by attacking the foreign nucleic acids based on their different sequences from self. And you can evolve new CRISPR-Cas sequences to attack a new bacteriophage when you're a bacteria. And even simple eukaryotes can evolve new sequences to attack invaders. But when it comes to multicellular organisms, you can't evolve that quickly. So new defenses evolved that are based on nucleic acid immune sensing receptors that don't attack the foreign nucleic acids directly, but they distinguish viral from self and can induce CD8 T cells then to kill infected cells. And the way they distinguish them is based both on the RNA being different, pseudouridine 2'-methyl, for example, and the DNA being different. And those are the fundamental differences that allow self-non-self discrimination and protecting genome integrity. So these are the receptors that I want to focus on for today, just at a high level. For distinguishing DNA, TLR9 is the key receptor, distinguishing unmethylated viral CpG versus the 5-methyl DNA in ourselves. Turns out, in addition to the agonist binding sites on TLR9, there's an antagonist site. It's not as well understood. We don't even have a crystal structure of human TLR9. The only crystal structure we have is mouse. But we do know that the TLR9 agonist is very good at inducing plasmacytoid dendritic cells, PDCs, to make interferon alpha. Now, the other DNA receptor is the cGAS-STING pathway. That's in the cytoplasm rather than the endosomes. And cGAS-STING does not distinguish self and non-self. In fact, it's typically activated when DNA leaks out of the endosome, out of the nucleus in stressed cells into the cytoplasm, and that can activate that pathway. cGAS-STING also induces type 1 interferons, but it's predominantly interferon beta rather than interferon alpha. And I'm going to spend some time talking about the differences among type 1 interferons, because I think that's not as broadly discussed as it should be. For RNA, there's multiple different receptors. Most of them distinguish double-stranded RNA. The two that are able to distinguish single-stranded RNA are 7 and 8, and that's based on guanidine-uridine-rich RNA and pseudouridine in self. And there is an antagonist site in both 7 and 8. That antagonist site has recently been reported to be activated by 2'-methyl guanosine. So you have multiple layers of self-non-self discrimination here. In the agonist binding site, you don't activate if you have pseudouridine. And if 2'-methyl guanosine is present, you turn it off anyway. So that allows a higher level of discrimination for self and non-self. And again, you can activate the PDC to make interferon alpha. This especially requires phosphodiester GU-rich RNA. When we initially published the first RNA sequences that are agonists for 7 and 8, more than 20 years ago, we were using a phosphorothioate backbone, and we've now realized that the processing in the endosomes requires nucleases like PLD3, PLD4, RNase. Those really work much better with a phosphodiester template. So the key point here is there's multiple different receptors. What's unique is the interferon alpha that is really only induced by 7 and 9 predominantly. Those are the two that are expressed in plasmacytoid dendritic cells. So they're unique, and I'm going to spend some more time talking about them. First, just a little bit more background on the type 1 interferon system. We have 17 type 1 interferons, 13 interferon alpha genes, interferon beta. They all bind to the same receptor. This IFNAR receptor is expressed in all nucleated cells in the body, so it's ubiquitous, and the downstream signaling pathways are reasonably well understood. So why do we need 13 genes for interferon alpha? I'm going to come back to that. But first, let me just give a little more background on how we know that type 1 interferons are important in tumor immunology. The most convincing early data came in mouse models where IFNAR was knocked out. And if you knock out IFNAR in mice, they're more tumor prone to carcinogen exposure. Those tumors are more immunogenic, actually, when they're transplanted into wild-type mice. And if you even just knock out IFNAR in dendritic cells, you lose the ability to cross-present tumor antigens. Once recombinant interferon beta and recombinant interferon alpha were produced, this is now decades ago, people started putting them into mouse models. And this is an early paper from 1992. But what you see actually is interferon beta down at the bottom here is better at inhibiting tumor growth than interferon alpha in mice. Key take-home message is alpha and beta both have anti-tumor properties in mice, and inducers of beta or alpha both have anti-tumor properties in mice. This has led to a widespread assumption among cancer immunobiologists that all type 1 interferons are the same. And one key message I want to give you today is they are not the same. Let's just start with this data that is just in bioRxiv right now. What they did was to take a couple of different recombinant interferons. These are several different recombinant interferon alphas, recombinant interferon beta, and omega. They incubated human PBMCs and then did single-cell RNA-seq. And just looking at the total number of mRNAs that are upregulated by each one, you see there's some unique genes with each. There's a lot of genes that are induced by all of the type 1 interferons, but unique ones too. Same with long non-coding RNAs. Number of genes downregulated, some are common to all, but there's also some unique. So each of these different interferon alphas are having different biologic effects. Now, how do we know that those are important? This is just transcriptional data. Well, there have been some investigators who've tried the different recombinant interferon alphas in viral models, viral replication models, and for inhibition of SARS-CoV or flu virus, HIV, hepatitis viruses. Each different interferon alpha has a different profile of which virus it inhibits best. So even though these all bind to the same receptor, there are slight differences in their on and off rate, exactly how they bind the two receptor subunits, alpha 1 and alpha 2. And that leads to different downstream effects that are biologically important. And so a conclusion of this, no single recombinant interferon alpha can possibly reproduce the complexity of all 13 genes being induced through the endogenous pathways that upregulate these. So for therapeutic purposes, I think we should take advantage of that. And I think the reason we have 13 genes that have been maintained independently through evolution is because that's what we need. We've been under intense selective pressure, as have all other vertebrates that have many of these genes, and they function. Now, some evidence that we have for the difference of alpha and beta are as soon as the recombinant interferons were made, they were put into human clinical trials in just all kinds of different diseases. And what fell out of that is that interferon beta failed repeatedly for treating hepatitis B, hepatitis C, cancers, where alpha was highly effective and is approved. But it succeeded in treating multiple sclerosis. Alpha did not work there. We know that the mechanism for multiple sclerosis is IL-10 and Treg induction by beta. So this is a clear biologic difference between interferon beta and interferon alpha. If we are to do cancer immunotherapy, I would submit that we need to focus on inducing interferon alpha, the highest ratio that we can of interferon alpha to beta, because the affinity of beta for IFNAR is 10 to 100 times higher. So if they're both being made at equivalent quantities, beta is going to dominate. We need to focus on inducing the highest levels of alpha that we can. So how do we do that? I already mentioned plasmacytoid dendritic cells are the source, in particular this subset called P1 plasmacytoid dendritic cells, which really are only activated by TLR7 and TLR9. Now, the CGAS sting pathway, as I mentioned, doesn't activate that. It's not distinguishing self-non-self, predominantly induces interferon beta. I think that a lot of people in the field are believing this may function more to signal cell stress, that something is wrong, recruit other immune cells to come in and figure out why this cell is stressed. Is it stressed just because it's hypoxic and you need to stimulate angiogenesis, or is it infected? TLR7 and 9 are the primary inducers for interferon alpha, and so that's what we should be trying to focus on. For TLR7, there's evidence already out there, a number of different programs that they have some function by themselves, and I was really glad that Daryl mentioned the Alicio program with the CPGB, because I think their data are really pretty impressive in minimal residual disease pancreatic cancer, and he also mentioned vidutolimod, the CPGA that we were developing in my last company, Checkmate. So what I want to address today is, why don't we try to combine these? And I will say, early on when we discovered the first RNAs activating TLR7, we did try combining them with CpG DNA, and we more often saw antagonistic effects than any kind of additive effect. We have now figured out how to combine those structures using phosphodiester DNA, and I'll walk you through that. So the three topics I want to cover now are, first, developing the CpG DNA agonist for TLR9, then I'll talk a little bit about TLR7/8 agonists, and then I'll talk about combining them together. So as you already know, I think in our DNA, CpGs are methylated. In viral DNA and bacterial DNA, they're not methylated. We know the mouse crystal structure for TLR9 and where the binding sites are, how CpG fits in there. If both of those sites are activated, you get the strongest activation. And this is very much dependent on the DNA backbone. Phosphorothioate backbones prevent the processing that allows the optimal binding of CpG DNA into those two different binding sites. This processing is mediated by DNase 2. If you knock out DNase 2, the cells have the same response to phosphorothioate and phosphodiester DNA. In the clinic, this is the first CpG DNA that we took into clinical trials back in 1999. It has an all-phosphorothioate backbone. This is in an approved vaccine now as an adjuvant. It's a very good adjuvant. It's great at activating B cells. It's not so strong at activating the plasmacytoid dendritic cell interferon response, and that's because of the backbone preventing the processing. This nevertheless went into clinical trials, including phase 3 oncology trials. It failed as a monotherapy, failed in combination therapy, and other CpG phosphorothioate backbone oligos. There were 2 CpG-C type oligos that went into clinical development. They also have failed to show monotherapy activity in oncology, at least so far. Some groups are still working on that. This is the type that, as Daryl mentioned, because of these poly-G sequences on the ends, this is very difficult to work with. These G structures form G quadruplexes, and we had to work out how to formulate these, how to control the aggregation. This is the palindrome in the backbone as well. It forms double-stranded DNA. But we were able to do this, and we did see monotherapy activity in advanced cancer, patients with PD-1 resistant melanoma. We had 9 out of 40 patients who did show a response just to this treatment by itself in a virus-like particle. Now, these quadruplexes are interesting because G quadruplexes are present in the genomes of all viruses and retroviruses. They're required for their life cycle. And so it's possible that the reason this works so much better, in addition to enabling the processing, maybe we're just more effectively mimicking the viral structure. I will add one point that I think is fascinating. Retroviruses have suppressed CpG expression in their genomes. So they only have about 15% of the CpGs that should be present if base usage was random. And that is consistent with evolutionary pressure to avoid triggering this pathway. So I think this is a pathway that we want to take advantage of for cancer immunotherapy. We already have some evidence that it works on its own, although I think we can improve on that. And here's how. This is what vidutolimod looks like. It's a virus-like particle. As a virus-like particle, it's very immunogenic, and so you get antibodies made by B cells after the first dose. And once those antibodies get up to a certain level, they start suppressing the interferon production by the plasmacytoid dendritic cell. The other thing that we learned in our trials was when patients had high levels of myeloid cells in their tumors, they didn't respond to vidutolimod. And we now know these cells suppress plasmacytoid dendritic cells unless they're activated through TLR7 or 8. These do not express TLR9 in humans. In mice, they do. Likewise, conventional dendritic cells, the various populations, express TLR7 or TLR8. And so this provides a rationale then to combine TLR7/8 activation with the TLR9 activation to try to overcome that suppression and to switch from the virus-like particle delivery that's immunogenic to a lipid nanoparticle delivery that's not immunogenic to avoid that. And the LNP also would look more like a virus. So let me talk now about RNA agonists. And let me start, first of all, with the small molecule agonists that are being widely developed today. Multiple companies are using small molecules such as R848 that bind the TLR7 homodimer you can see here at just this first site. And we've known this since, I think it was 2016 when this site was when this was published. That binding at that first site, it causes a conformational shift in TLR7 that activates the NF-kappa B pathway. And so if you look in standard reporter cells you can buy from Invivagen, R848 is a very strong stimulator of that pathway. Now, when we package a phosphodiester GU-rich RNA into a lipid nanoparticle and we try to activate this, we don't get a signal. Why is that? Oh, and I was just going to mention, R848 is in development. ICON is one of the companies developing this there in the Bay Area. Roger Perlmutter, who was formerly head of R&D at Merck, is their CEO. It has a narrow therapeutic index, and it does seem to have some efficacy in the clinic, but it's modest. So the difference with RNA is it binds both sites. The second site here prefers UUU. Guanosine binds the first site that also binds the R848. This causes a different allosteric shift, and it results in activating the IRF pathway. So the signal that you get, you can't pick up in these reporter cell assays. You have to use a PBMC assay for the interferon alpha signaling. R848 induces a little bit of interferon alpha, but it's nothing like this. So it's not a case of one being a stronger activator than another. It's a different allosteric shift that turns off the NF-kappa B pathway, turns on the IRF pathway. Both of those are downstream from TLR7. The nature of the agonist that you provide determines what the balance of those two pathways is. And similar mechanisms appear to operate with TLR8 and TLR9. So we want to combine these. And for TLR7/8, we're going to use phosphodiester backbone GU-rich RNA. For the TLR9, we're using the CpGA DNA. And we just make a single oligo where we have an RNA domain and a DNA domain. The oligo that we're planning to take forward into the clinic is less than 40 bases long, and it's designed to self-assemble so that it can easily be packaged into an LNP. We screened a lot of different lipids for our LNP formulations, and what we were selecting for was low inflammation. We were looking for LNP designs that did not induce TNF or IL-6 or IL-1 beta, and that gave us the highest possible interferon alpha. And we were also selecting for lipids that had already been in humans previously. One other point I should make. Because the TLRs are in endosomes, we don't need ionizable lipids that are usually used in LNPs to lyse endosomes and allow endosomal escape into the cytoplasm. If you're delivering an mRNA or an siRNA or a CRISPR guide RNA, you have to get that out of the endosome into the cytoplasm, and that requires ionizable lipids. We don't need ionizable lipids for this, so we're actually not using them in our LNP. So when you combine these together, DNA by itself, in this experiment at least, has a modest effect for inducing interferon alpha. The RNA was a little stronger. And when we combine them together, we get the best results yet. And this is just adding 0.5 micrograms per mil of the DNA, this level here, to increasing amounts of the RNA here. So there is some cooperativity there, and this occurs without inducing very much IL-6. This is IL-6 induction by R848 as a positive control. So coming back to tumors now, and how do we develop this for cancer immunotherapy? I think it's well understood that tumors are programming myeloid immune suppression by releasing damage signals. And those damage signals are often packaged in exosomes that are taken up through lipid receptors on the surface of the myeloid cells. And that programs those cells for the wound healing response. There's no infection in this tissue. These cells are detecting damage. They're trying to promote angiogenesis and heal the tumor. So what we want to do now is to change that program by introducing essentially a synthetic retrovirus with a lipid nanoparticle containing the DNA to activate TLR9, the RNA to activate TLR7 and TLR8 in all of these different immune cell populations. And together, if all those immune cell populations are activated in the right way, our hypothesis was that we might reprogram this. We might actually change the transcriptional programs of these cells from the wound healing response to the antiviral response. And again, the LNP enables uptake via lipid receptors that are already present on these immune cells. One example that's been well studied in the literature is CD36. So does this actually work? We took human tumor immune cells. These are from ascites fluids in patients with abdominal tumors. This work was done by our collaborators at Iowa. And then you just culture those ascites cells with either our lipid nanoparticle, or this is just a CpG-A DNA that we were developing at Checkmate and now with Regeneron. And then this is the ICON payload, the R848. So for interferon alpha, we induce a lot of it. The inflammatory cytokines that you really don't want, we're not inducing, whereas R848 is. And so this again shows the difference, at least at a cytokine induction level, between an RNA DNA agonist and the previous generations of agonists that were available. But to show transcriptional reprogramming, we used RNA-seq. And so we performed RNA-seq on these cells. And the signatures that I think are most intriguing for this purpose are signatures that were recently published as distinguishing responders to immune checkpoint inhibition from non-responders among patients who had colorectal cancer. So patients whose baseline biopsies showed strong signatures for interferon alpha, this is a 25 gene signature, this is a 60 gene signature, the interferon gamma, a CXCL10 signature, and of course M1 signatures have been around for a while. Higher levels of that predicted or were associated with response to checkpoint inhibitors. And so you can see the untreated cells have comparatively low levels. R848 does induce those signatures a bit, but the Z-007, our triple agonist, induces them to a higher degree. We also looked at sort of bad signatures that are associated with immune suppression. Hypoxia, this is a signature for myeloid-derived suppressor cells. And R848 actually induced those bad signals too, showing more, just the difference in the biology here, whereas they're reduced by our drug. Now, this is just one patient. We need to repeat this in a lot more patients. But if we can do this in vivo, this suggests that we might actually be able to reprogram the tumor microenvironment. And colorectal cancer, with liver metastases, that's one of the big unmet medical needs for cancer immunotherapy. So we think that the failures of TLR agonists in past clinical development may not apply to what we're trying to do now. And we've seen the same signatures not only in the human ascites cells. This is a mouse liver metastasis model that I'll show you in the next slide at Miriam Murad's lab in Mount Sinai, where the Z-007 treatment in this model induces those same signatures. And this is healthy monkeys, where we also looked in the liver and the spleen and lymph nodes of the monkeys. We see the same signatures induced there. Now it's transient. This is the control. This is at 4 hours after Z-007, so it's a pretty rapid induction of the signature. But a week later, it's back down to baseline again. So this is not a permanent state. So this is the model that Miriam Murad's lab, Jesse Bumelha in the lab has been running. It's a quadruple mutant model where you get organoids that are implanted into recipient mice to transmit the tumor, and then give injections of Z-007 here. And then the mice were killed, I think it's 6 days after the last treatment. There was reduction in the size of the tumor, increase in myeloid cells in the tumor microenvironment, increase in CD8 T cells, and in interferon alpha secreting plasmacytodendritic cells. So all what we want to see. Another model in TRAMP, this is a prostate cancer model run at Hayden Kissick's lab in Emory. And in this model, he looked at treatment, intratumoral injection, with or without anti-PD-L1. And you can see the effects of them individually and in combination, a little bit better reduction in tumor growth. What's intriguing in terms of the granzyme B positive T cells, you can really see the contribution of the TLR7 789 agonist in inducing those cells where the checkpoint inhibitor doesn't. And the combination is the best of all. Another model in mice, MC38, we see reduction in tumor growth. And in fact, at the high dose of 10 micrograms, 8 of 8 mice cleared their tumors. At the 2 microgram dose, 5 of 8 mice did. The mice gained weight normally during the study, so we didn't see any toxicity from that compared to 5FU, which does interfere with weight gain. This isn't a particularly difficult mouse model, and really none of the mouse models have predicted human results. So I'm more excited about the results from a pilot study in pet dogs with cancer. Pet dogs develop spontaneous tumors. About a quarter of all pet dogs will develop cancer. And because they share our living environment, they're exposed to the same factors, and their tumors are just much more similar to human tumors. So in a pilot study, we treated 7 pet dogs at a veterinary clinic. 3 of those dogs have shown at least a 30% reduction in tumor mass. And I'm just going to focus on Guero, who had a metastatic hemangiosarcoma. He only received treatment with Z-007. He had previously failed to respond to Palladia, a tyrosine kinase inhibitor. At baseline, he had 13 metastases in the lung. They all cleared after a few months. Skin lesions cleared. And a metastasis in the heart lost about 90% of its volume during treatment. And throughout this treatment, over 8 months, he had no toxicity from treatment. He didn't have fever, diarrhea, vomiting, the other kinds of things that you worry about typically. Also, no change in liver function, kidney function, really did amazingly well through that. More recently, we started compassionate use in several other pet dogs, and a second dog with hemangiosarcoma also now is doing incredibly well. They're only about 6 weeks in, so hopefully that will persist. What about safety? In monkeys, this is the equivalent dose that we were using in the dogs. I'll just go back here. So Guerrero received between 50 micrograms to 200 micrograms weekly, and he was 20 kilos. These monkeys were about 4 to 5 kilos. So a 20 microgram dose in a monkey is the equivalent dose in the dogs. At that dose, in terms of serum response, we see a little bit of interferon alpha in the serum, more CXCL10. That's the biomarker that we've used in the past in vivo. Because in the past, with other TLR agonists, we never really saw much interferon alpha induction at any dose in humans or monkeys. As we get up to higher doses in the monkeys, we're seeing a lot of interferon alpha. And we're seeing much higher levels of CXCL10 also. So this is over 100-fold dose range. Over that 100-fold dose range, we did not see toxicity in the monkeys. They didn't have fever. They didn't have liver enzyme elevations. So for the first time, I think, we have an innate immune agonist that dissociates the cytokine release syndrome that we used to think of as being an essential part of immunotherapy. It's possible to induce very high levels of interferon alpha and CXCL10 without that cytokine storm. And I think that's really exciting for the potential applications in humans. I just want to make one other point about these data. When we think about CXCL10, in our prior clinical trials in humans with the TLR9 agonist, responders had increases in CXCL10 that were higher than non-responders on average. The highest responses we got were between about 5 to 10,000 picograms per mil. So that's the level that we're shooting for in humans, and we achieve that in monkeys, at least at this very low dose. Now, we may have to go up to much higher doses in humans, but I think this is very encouraging for where we're at in development. So let me just take a step back now and try to integrate this at a high level with how I think we should think about innate immunity. Innate immunity evolved to distinguish these three different states: intracellular infection, extracellular infection, and cell damage, sterile cell damage. Pattern recognition receptors are how immune cells compute those differences. And we have all these different pattern recognition receptor families, the TLRs, the NLRs, the RLRs, and so forth. And somehow the net result of those inputs allows the immune system most of the time to make an appropriate response. What I would submit is that for retrovirus, as the case of the key intracellular infection, that once a retrovirus has infected your cell, the only way you're eradicating that infection is by killing that cell. And so CD8 T cells have that job. CD8 T cells can't detect the retrovirus. They don't have any TLRs. It's these other immune cell populations that are responsible for determining what's going on and then telling the T cell what to do. What type of cell do you kill? And so the minimal signals that are used, that evolved to detect a retrovirus, are the single-stranded RNA receptors, TLR7 and TLR8, and the CpG DNA receptor, TLR9. That had to evolve to be sufficient to activate CD8 T cells in the absence of other signals and to induce that response. This is what we have to accomplish for cancer immunotherapy. And I think combining those three receptors should become a core part of cancer immunotherapy combination regimens. Extracellular infections are a lot easier to detect. There's a whole lot of different receptors for those molecules. I won't go into those. And then we have cell damage. And of course, I'm showing here exosomes being released. When immune cells come into tumors or inflamed tissues, they upregulate their lipid uptake receptors so that they can taste what's going on in those cells. And if it really is just sterile tissue damage, angiogenesis is the appropriate response, the wound repair response that I talked about before. These cells suppress T cells. Tumors, of course, are mimicking damage. They are triggering that response. And so what we want to do is to reprogram that. I think from the data that we have so far, I think it may be possible actually to reprogram this in a living patient using the right combination of agents. And maybe we'll need to add checkpoint inhibitors as well to further improve the efficacy of those CD8 T cells. But I think we need to get this into humans to determine that. So the conclusions that hopefully I've convinced you of, CGAS sting agent agonists are great for inducing interferon beta, not so much for inducing interferon alpha. They work great in mice because interferon beta has anti-tumor activity in mice. It does not in humans. Sting agonists, of course, have failed repeatedly in human clinical trials. And I don't expect them to all of a sudden start succeeding, no matter what you do with them. On the other hand, TLR789 agonists do what you want for an innate immune agonist in treating cancer. We've shown in the data here, we can reprogram tumor-associated immune cells, inducing the signatures that distinguish responders from non-responders to checkpoint inhibitors. We've shown in vivo safety and efficacy in these various models. And the non-immunogenic LNP delivery avoids inflammatory cytokines and the anti-vector response. So we plan to pursue the clinical development of Z-007. We're currently doing the GMP manufacturing, and we just got feedback from the FDA. They're going to allow us to do single species GLP safety, which makes it cheaper and faster for us to get into the clinic. And they approved our plans for dose escalation in humans. So we hope we'll be in the clinic early next year. So just wanted to acknowledge the collaborators and our team, both within Zola and our various CROs and our academic collaborators as well. And part of my goal in reaching out, we are a virtual company. We don't have any of our own offices or labs, so all the work is done via collaborators, and we're certainly interested in talking to new collaborators